We are back again with a new topic, separation techniques. First one is the centrifugation. Separation techniques, principles and applications of the centrifugation. Centrifugation is a technique of separation of substances which involves the applications of centrifugation, or the particles get separated from the solution of the solution according to their size, shape, density and viscosity of the medium, and the rotating speed, okay, and through which it is getting separated. Now the separation is so very important, and it has lots and lots of significant role in the chemical industries, in the pharmaceutical industries and many more places where we could segregate them into different particles. We have many other physical separation techniques also, like hand picking, threshing, winnowing, sieving, evaporation, distillation, filtration, many more, sedimentation, everything we have. But here we are going to segregate certain kinds of liquids from the liquids, okay, liquid to liquid, or it is of biomolecules, in other words, if I have to say you all, the supernatant and the residue what we get after the centrifugation. So here the position, the particles, those density is lighter than that of the solvent, which will sink, and the other particles that are lighter than that of the other one or the solvent will start floating on the top. The greater differences in the density, the faster is this particular mechanism. So if two different kinds of densities, substances or the solvent and the solution, if we are taking, if you are mixing it together and if you are trying to separate it out, it is very much easy to separate it if their densities are different from each other. Okay, so the centrifuge works using the sedimentation principle, where the centripetal acceleration carries the denser substances and the particles more downwards, and in the radial directions. Okay, so the types of centrifugation, let us go through, and their applications we will study a little later. So the differential pelleting is one of the first method of the centrifugation. What are we going to do with this? How are we actually going to go about this? Is the tissue such as the liver, homogenizing that, and at 32 degrees Celsius in a sucrose solution, and it also contains that of the buffer. The homogenate is then placed in a centrifuge and spin at constant centrifugal force at a constant temperature. Then you can see that after some time there is a sediment formation at the bottom of the centrifuge called as pellet, and overlining the solution is the supernatant. Okay, so the complete separation is going like this, as the picture saying. You see all the mixture of solutions are present in this particular test tube. As we have done first step of centrifugation, we could see that more denser particles, that is the bigger green color particles than that of the red and blue, have got settled first. Okay, so once they have got settled, we will remove the supernatant part, that is the first liquid part, and the pellet part is thrown out. Then, taking that into the consideration, again you go to another round of a centrifugation, you would find the red color spots are more heavier or denser than that of the blue and black. Then the red is going to settle down because they are much heavier than them. Then the next step, you could see that you take the supernatant of blue and black dots or the pellets, then you would see that blue color pellets are completely separated or got sedimented down. Okay, removal of the supernatant, you get the black color pellets, and the blue color pellets remains in the other test tube. So here the four different kinds of molecules are getting separated by the centrifugation methods, but it's a process, step by process. Every from one step all the four cannot be segregated, that is there, but yes, you can separate it stepwise. So the particles then travel through the gradient until they reach a point at which their density matches with the density of the surroundings. So there, till there the the rotations is going on and the settlement will be trying to happen. Thereby the fraction is removed and the analyze, and it will be analyzed. Okay, and see here, next one is the density gradient centrifugation. This type of centrifugation is mainly used to purify various ribosomes, membranes, etc. The sucrose density gradient is created gently overlapping lower concentrations of the sucrose on the higher concentrations of the centrifuge tubes. The particles of interest are placed on top of the gradient and centrifuge in ultracentrifuge. The particles travel through the gradient until they reach reach a point that their density matches the density of the surrounding sucrose. So here also it's the same thing that of the other one, but here it is again a density or a mass gradient is going to be acting on it, and the fraction is removed and it will be analyzed. So here it's again the same thing. So when you centrifuge it, different grades at different gradient, they are going to get settled down. Here there before it was a mixture, okay, only two solutions was getting separated, but here you could see that there are three separate zones or the gradient regions that you could see that it has got separated into. Okay, there's a slight difference between this and that. See if you can see that it is see uh, the the higher concentration of the sucrose is making them uh, get into different zones of the uh, zones and get settled down. Okay, this is a kind of a density gradient centrifugation wherein we can immediately segregate all the three parts. Rate zonal density gradient centrifugation. What is this rate zonal density gradation? See, again you can see the picture here. I'll explain you what that is. See, the zonal centrifugation is also known as the band or a gradient centrifugation, same that of the density gradient centrifugation. It relies on the concept of the sedimentation coefficient, that is movement of sedimentation through the liquid medium. It is a technique, density gradient, created in a test tube which sucrose and high density at the bottom. The sample of the protein placed at the top of the gradient and the centrifugation is done. See, with the centrifugation, sedimentation is faster. Their particles in the sample are move ahead of the slower, and that is that is some separated particles you can see, and they are into different zones and its gradients. The coefficient and the fraction are affected. Okay, and by end of the centrifugation you could find that there is a zones of zones of molecules being formed, and here you could see that there is a density gradient. See, different as the time passes you could find that there is a complete separation of the molecules. Slowly they are getting into a different rate zonal gradient regions. Okay, so next is the isopynic centrifugation, whereas here the sample is loaded into the test tube with a gradient forming the solution. The solution of the biological sample is taken here, maybe some of the eggs you can say. See all the different types of eggs or zonal A, B, and C. Here, this is also similar to that of the zonal gradients, but what is the difference here is the density of the solutions is more or less similar. Okay, so here under the influence of the centrifugation force, the cesium or the cells are getting settled down in a different density gradient from the top to the bottom. Particles from a different point, they are moving to a different grades of forms the different gradients. But here one thing that you'll have to remember is the buoyancy plays an important role. Okay, where a particular substance starts floating, at which particular zone that the particles or the material starts floating or the buoyancy is seen, there the density buoyant density equals that the part of the gradient, and there's a formation of the bands. This is to say that the sample molecules move to the region where the density equals the density gradient, and it is true equilibrium procedure since depends on the buoyancy density. Okay, buoyant density, and there is no velocity is seen here. Okay, only depending on the buoyancy they are going to segregate themselves and form a different zonal regions. Okay, so here is the zone A, B, and C, which you can see, which is an isopynic centrifugation. So applications, if you go on, then to separate two miscible substances which have got completely mixed up with one another, to analyze the hydrodynamic properties of the macromolecules and purification of the animal cells or the mammalian cells. And you have fractionalization or fractionation of the subcellular organelles of the membrane vesicles, removal fat from the from the uh, from the milk to produce skimmed milk, and separating particles from an air flow using the cyclonic separations and classifications, and the stabilization of different wines, etc., and separation of urine components and the blood components in the forensic sciences and the research laboratories. And also it has been used as an aid in separation of the proteins using the purification techniques, such as you have many other techniques like separating out the ammonium sulfate and its purifications. Okay, so as we have studied the separation techniques, different separation techniques of the centrifugation only. So centrifugation is going to play a very important role, as our application says. So here ends up the topic of the separation technique of the centrifugation. In the next video we'll be coming up with the separation technique through the chromatography. In that there are different types and its applications we will be studying. Okay, see you soon. If you have liked the video, subscribe for it and like it. Okay, thank you.